So yeah, my name's Laura Branscombe. I'm currently in my third year of my PhD, and I'm based down in the southwest of England in Plymouth. So I'm joint with the Marine Biological Association and the University of Plymouth. My PhD has largely been focused on looking at how diatoms, so these single-celled algae, interact with other marine microorganisms. And today I'll be talking to you about the diversity and seasonal patterns of some of these interactions in the Western English Channel. Sorry, has that changed for everyone?
Yes.
Perfect. So before I came to the Marine Biological Association, I was a marine biologist and then a microbiologist, but I'd never actually sort of studied diatoms. So I'll just give you a little bit of background to start with. So diatoms are a major group of photosynthetic microalgae. So they're these really beautiful single-celled photosynthetic microorganisms that you can see here in this picture on the right. So they're really, really diverse. They've got a diverse range of forms, and they're made of these sort of silica-based cell walls. So they form these really beautiful, intricate sort of glass shells almost. And they're really dominant in both marine and freshwater phytoplankton communities. So because of their ubiquity and abundance in the oceans in particular, they're actually responsible for around 40% of all marine primary productivity. So 40% of all the oxygen produced by photosynthetic microorganisms in the oceans is produced by diatoms. And so you might have heard of diatoms in some capacity in the news. So they can form these large and sometimes toxic blooms. So they can often be the causative agents for harmful algal blooms. And this picture again on the right just shows a sort of massive bloom of a genus of diatom, so Pseudonychia, off the coast of Washington. And these blooms are so large that they can quite often be caught on satellite images. And because of the sort of size of these blooms and the sort of dominance of diatoms in marine communities, they can have really significant impact on marine carbon cycles and also marine nitrogen and silica cycles. Not only that, but these diatom communities and these sort of blooms in particular can also really impact marine phytoplankton communities. So they have sort of wide-reaching impacts on not only other marine microorganisms, but also on sort of hydrotrophic levels. So they can impact things such as marine mammals, marine birds, particularly these toxic blooms. They can sort of accumulate in coastal regions, in shellfish, and they can really significantly impact aquaculture industries. So you might have heard of sort of mussel farms being shut down because of these large toxic blooms, and all of these toxins are sort of concentrating within the mussels, within these filter-feeding organisms. So that's just sort of a brief bit of background, but hopefully it's enough to convince you that diatoms are not only these sort of beautiful, intricate microalgae, but also they're really significant in marine communities. As I said, diatoms don't sort of exist in isolation. They exist in these planktonic communities. So they interact with other microorganisms constantly. And because of this, they've formed this really complex web of interactions with other planktonic microorganisms, such as marine fungi, marine bacteria, marine viruses, and also other zooplankton, so such as copepods and things like that. These interactions are really complex. They can impact diatom growth, physiology, and even their toxicity. So there's been quite a lot of individual studies about how these organisms interact and the sort of chemicals which mediate these interactions. So quite often these interactions between diatoms and other microorganisms will be mediated by chemicals, metabolites that the diatoms or the other microorganisms are sort of putting out into the water column. I will come on to this in a little bit more detail later, and if you have any questions at the end, feel free to let me know. But yeah, so as two of the major groups within the phytoplankton, diatoms and bacteria, as you might have guessed, are what I'm most interested in. Again, diatoms and bacteria also have this really complex web of interactions, which also influence diatom growth and physiology. So diatoms interact with bacteria all the way from synergistic bacteria, so interactions with bacteria which provide the diatoms with nutrients such as vitamins, which they might not be able to provide for themselves, and the diatoms in return will give the bacteria sort of dissolved organic carbon. And then these interactions range all the way through to antagonistic interactions, for example, with algaecidal bacteria. And as it was probably quite obvious from my title, that's what I'm most interested in. So these interactions between diatoms and algaecidal bacteria, so these antagonistic interactions. So these algaecidal bacteria are really, really important. They, again, have impacts on diatom growth and physiology, and they've also been linked to sort of diatom bloom termination. So when you see these large bloom events sort of in coastal regions, whether they're toxic or not, algaecidal bacteria will sort of often come along at the end and sort of lyse all of these diatoms, and will cause this sort of mass cell death of all of these diatoms. And this releases a massive sort of flux of carbon into the marine environment, which really significantly impacts sort of phytoplankton communities. Naturally, algaecidal bacteria are of quite a bit of interest, so there's quite a lot of studies that have been conducted into interactions between diatoms and algaecidal bacteria. Unfortunately, these studies tend to be sort of two-component systems, so it will be a case of someone researching one particular diatom and one particular algaecidal bacterium, and how they interact in the lab and what is sort of going on with the physiology of both of these organisms. However, we don't really know the diversity, abundance, and ecological significance of these algaecidal bacteria in nature, or of these antagonistic diatom bacteria interactions in nature. And we think this is because of a lack of routine sampling. So we know that these algaecidal bacteria exist. We know in some cases what they do to the diatom, what the diatom affects on the bacteria, but we don't really know much about how they're interacting in the natural environment, and even how many of them, how diverse they are, and these sorts of things. So that's where my PhD comes in, to sort of get a little bit of a better understanding of this massive knowledge gap. I had to start with, I had one main aim, which was to explore the diversity and abundance of these antagonistic diatom bacteria interactions in the Western English Channel. Through doing this, so as I said, I'm in my third year, so I've been doing this for quite a while. Through doing this, I've isolated a particular algaecidal bacterium called Pontococcus alexandrii, and I want to investigate the algaecidal mechanism of this bacteria with a range of bloom-forming diatoms. And then to further that, I want to look at the ecological significance of this algaecidal bacterium with these diatoms. So how widespread is this? These interactions, are they really ecologically significant? So luckily for me, I'm based in Plymouth, right down in the southwest, and that's not only lucky because we have views like this, but it also means that I have really, really easy access to the Western Channel Observatory. So the Western Channel Observatory is this sort of large marine station just off the coast of Plymouth and Cornwall. And it's really regularly monitored by the Marine Biological Association, by Plymouth Marine Lab and the University of Plymouth. So we have this sort of routine sampling, not only by these research vessels, so our research vessel and the research vessels of PML, but also these sort of autonomous buoys. So the Western Channel Observatory is really, really well characterised. We know a lot about the sort of microbial communities that live there. We know a lot about the environmental conditions, so nutrient levels, temperature, salinity, oxygen, things like that. And that's sort of available to us weekly. So this is really, really handy for us if we want to do any studies such as this. We have all of the sort of metadata to accompany it. And so for this reason, I've chosen the Western Channel Observatory as my model sampling station. So L4 station, just off the coast, so in the Western English Channel, is my model sampling station. It sees regular diatom spring blooms. So it's a really highly productive coastal region. We see these spring blooms there all the time. The microbial communities are well characterized, and we have all of this metadata. So it's the perfect place to sort of start looking for these algaecidal or antagonistic interactions. And as I said, so the sort of background, so the studies of the environmental relevance, the ecological significance of these interactions in nature is really, really poorly understood. And because of this, there are very few methods which we could actually sort of apply to look at these interactions. So the first aim of my first aim was to establish a robust plaque assay method, which would allow me to isolate some of these algacidal bacteria and get a feel for the diversity and abundance of them in the Western English Channel. So the first thing we did was we took seawater samples from L4 station. We filtered out all the eukaryotes, all the algae, zooplankton, all the fungi and things like that. And we concentrated the bacteria right down, so we had a really dense culture of all of the bacteria present at L4. We then took 5 different diatom host species. So these are 4 ecologically relevant, often bloom-forming diatom species, and our model species, Diatoms and Bacteria. And we inoculated this really dense bacterial culture into these 5 host species. We then mixed this diatom bacteria co-culture with agar, so semi-solid agar, and we plated them out onto agar plates. And the idea is that the diatoms will grow within the agar. They'll form this really nice, thick, confluent lawn of diatoms. And wherever algacidal bacteria are present, they'll either prevent the diatoms from growing or they'll actively lyse the diatoms. So you get these sort of zones of clearing, or what we call plaques. And within that, there are no diatoms, but you can get this really dense culture of your algacidal bacterium. This is great because then you can pick this, resuspend it in seawater, sterile seawater, and you can do 2 things with this. So you can re-inoculate this into a subsequent plaque assay to confirm your algacidal activity, and you can also streak it out onto agar and then obtain your bacterium in pure culture, which you can sequence and get a feel for who you're working with. So once I'd established this plaque assay method, my next task was to get a look at the diversity and abundance of these bacteria, these algacidal bacteria, over the course of a 12-month cycle. So this was quite a big task for just a single person, but what I wanted to do was take a look at these seawater samples, so every other week. So every other week I would get seawater samples, I would take the bacteria, I would conduct these plaque assays, and I would see what sort of organisms we were getting over the course of a 12-month cycle. So this would give us sort of a picture of the seasonal patterns and the diversity as well. This worked really, really well. We conducted this for 12, 13 months. And we got a really nice diversity, not only of bacteria, but also sort of plaque formation. And again, we got these on all different types of diatom host species, which was also really convincing for us. And so we isolated the bacteria in pure culture, we sequenced them, and then we could get a feel for who we were working with, which bacteria we were working with. And this was again really, really nice because this gave us this showed us that we'd isolated algacidal or plaque-forming bacteria from a really diverse range of bacterial lineages. So alpha proteobacteria, gamma proteobacteria, firmicutes. It was really, really nice. And again, all of these bacteria isolated from a range of different diatom host species. So it might not be particularly obvious from this graph, but one thing that we did notice when we were conducting this is that we were getting, I mean, you might have guessed from the sort of aim slide, but we were seeing Pontococcus alexandrii pop up time and time again. So almost every month that we had plaque formation over this 12-month time series, we saw Pontococcus alexandrii pop up and cause these plaques on a range of different diatom host species. Obviously, this was really interesting for us. Pontococcus alexandrii isn't something that's typically been described as algacidal in the literature. To be honest, there's not really a whole lot in the literature that we can sort of infer about this, but we were seeing it time and time again, so we decided this seasonally persistent bacterium is quite interesting to us and maybe has some sort of ecological significance. So we decided to take that on and do some physiological experiments. And the first thing we did, we took the plaque, we streaked it out, and we also reinfected a subsequent assay. So we know Pontococcus alexandrii is this seasonally persistent, broad-ranged algaecidal bacterium that we find at L4 station. We then had it in pure culture. It was really great. We decided that we were going to do just some really basic physiological experiments. So does it cause sort of bloom termination? Does it cause this crash in a liquid culture? Obviously, all the work that we'd done so far had been in these sort of plaque assays, so they weren't necessarily the most environmentally relevant environments for this sort of interaction. Have a look at the photosynthetic efficiency and things like that. And then, unfortunately for me, Pontococcus alexandrii decided that it didn't want to be part of these experiments anymore. It stopped being algaecidal. And this left us scratching our heads quite a little bit. I spent quite a few months trying to figure out what had caused this sudden lack of algaecidal activity. I tried to rule out all of the environmental conditions, all of the culture conditions. I questioned whether this algaecidal activity had been contained on a plasmid that had been lost in cultivation. And it really was looking like this story was just going to go nowhere. Fortunately for me, the very last thing I tried was cultivating it on sort of maintaining this algaecidal bacterium on a culture of diatoms. So really simply, I just autoclaved some diatoms in agar, I plated them out, and then I streaked my bacteria onto this, what we call dead diatom media. And this then induced this really sort of big spike of algaecidal activity. So from this, we've managed to infer that Pontococcus alexandrii really only seems to exhibit its algaecidal activity when it's grown in the presence of its diatom host. This isn't necessarily a novel result. We have sort of seen these sorts of algaecidal switches in other bacteria, in other algaecidal bacteria. There seems to be sort of something about the old and dead diatoms that sort of does induce this algaecidal activity, which does make sense. But unfortunately, as I said, nothing much is known in the literature about Pontococcus alexandrii. So we have a few couple of steps that we want to try and to move on to. So we have really only started to dig into this algaecidal mechanism. And what we want to do next is to look at how and when this algaecidal activity is induced. So Pontococcus alexandrii is a rhoseobacter, I believe, and it has it has no genome associated with it, so the genome of Pontococcus alexandrii doesn't exist. This is something that we've done. We've had the genome of this bacterium sequenced, so that hopefully we can search through the genome and look for homologs of other well-known rhoseobacter or algaecidal sort of biosynthetic gene clusters. And what we want to do to complement this is a transcriptome analysis. So as I said, the induction of algaecidal activity in Pontococcus alexandrii is really sort of bolstered by this dead diatom media. So we've designed a really simple transcriptome study. We're going to go Pontococcus alexandrii on the media which doesn't induce activity and which does induce activity, and just see which genes are up and down regulated. And hopefully this will be able to inform our genome search. And hopefully with the transcriptome and genomes together, we'll be able to really put together this sort of algaecidal biosynthetic gene cluster within Pontococcus alexandrii. And we'll be able to get a bit of a story of when it's induced, how it's induced, potentially what algal compounds are being secreted that induce this activity. So what diatom signals are being given off to sort of kickstart this activity. Sorry about that. And then the next thing, so we have this potentially really nice story about Pontococcus alexandrii and the induction of its algaecidal activity. But as I mentioned at the beginning, one of the biggest knowledge gaps within the diatom bacteria interaction sort of field is that we don't know much about the ecological significance of these interactions. So from the data that I've collected so far, we believe that Pontococcus alexandrii is seasonally persistent at L4 in particular, at this really highly productive coastal region. But we don't know whether this is particularly abundant. We've isolated it a few times, but that doesn't necessarily mean that it's there in high numbers, and it doesn't necessarily mean that it's interacting with these diatoms at L4. So what we've been doing alongside of this is taking additional seawater samples and just getting a feel for the bulk assemblage. So we've been taking 16 and 18S amplicon sequencing samples. So what we'll do is sequence all of the bacteria and all of the diatoms at L4 station. And we'll do this over the course of 12 months again, so that we should be able to see really nice diatom peaks. So when the diatoms bloom, we should get a really big abundance of diatoms. And then we can also see if we can overlay the abundance of these bacteria with these sort of diatom bloom peaks. Unfortunately, we don't have any of the data back from this sort of section yet, so I can't say whether this is an ecologically significant interaction, but hopefully the next time that I give this talk, I'll be able to have a few more answers for you. And then the last thing that I want to do is look at the global distribution of Pontococcus alexandrii. So from what we see at L4, this seems to be this sort of coastal algal associated bacterium. But does this have a global distribution? Is this present in perhaps other coastal regions which also see diatom blooms? So all that I know currently from the literature about Pontococcus alexandrii is that it's commonly associated with a different type of microalgae called dinoflagellates. Unfortunately, it's not algaecidal against dinoflagellates, or there's nothing in the literature to suggest that it is. But are we seeing this bacterium present in other sort of diatom bloom regions, in other highly productive coastal regions? And to do this, I'm going to use databases, so these widely available databases such as the Tara Oceans database, Ocean Sampling Day database, just to sort of plug in my Pontococcus alexandrii gene sequence and see if we can find this in other locations. And so the take-home messages that I really want you to take from this are that we've managed to develop this plaque assay method, this really robust plaque assay method, which allows the isolation of a really diverse range of algaecidal bacteria. This fills, or could potentially fill, quite a large knowledge gap because this just isn't being done in other coastal regions or in other regions. Full stop. So this really does have potential application in other high activity bloom areas, and it can be used to systematically review antagonistic diatom bacteria interactions in nature. And hopefully people can take this forward, apply this in their sort of own regions, and get a little bit of a global picture of the scale and breadth of these algaecidal bacteria in nature. And then the second thing is that Pontococcus alexandrii seems to be a seasonally persistent and broad range algaecidal bacterium that we find at L4 station. And the algaecidal mechanism of this bacterium is a little bit complex, it's a little bit tricky at the moment. It does seem to be that the algaecidal activity is only induced in the presence of its diatom host. And hopefully, as I said, next time I'll be able to give you a few more answers about this. So thank you everyone for listening, and if you have any questions, please feel free to either get in touch via the chat, or my email is also down here at the bottom.